Hey everyone, welcome back. Uh, in our last video, we ran FastQC on our raw sequencing data, and sometimes FastQC gives you a beautiful green check mark, but often it gives you a big red X warning that your reads have low quality tails or are contaminated with artificial Illumina adapter sequences. So we cannot align contaminated data to a human genome, right? So it will cause massive errors. And a few videos ago, we also wrote a custom Python loop to trim quality scores, right? And it was too slow for so you have like 50 million reads, and it did not look for adapter sequences. So today we are bringing in the heavy artillery. We are going to use a professional command line tool called Cutadapt. It is blazing fast and industry standard, and the best part, it is actually written in Python, so which aligns with our current course as well. So let's jump into VS Code terminal and clean our data. Okay, so because Cutadapt it is a Python-based tool, installing it is incredibly simple. You don't need complex package managers. We can just use our old friend pip. Okay, so since I have it already installed, so this command will not will just tell me that it already exists, but I will just run it again so that you can see and copy. So if you run this command, okay, so it installed it. I think it got reinstalled or maybe it got updated. I don't know. So okay, once that is done, we will verify that it is installed correctly by asking for its version. Okay, so let's check it. Cut adapt version. And yes, if you see a version number like this pop up, you are ready to process some real genomic data. Okay, so when we run a professional trimmer, we are usually trying to accomplish two things simultaneously. First one is adapter trimming. So adapters are basically chemical handles glued to your DNA so the sequencing machine can grab them. If the machine reads past your DNA, it accidentally sequences the handles. So we need to cut it off. Okay, and second is quality trimming. As we saw in FastQC, the ends of the reads degrade. Okay, so we want to cut off any bases that have a Phred score below 20. Okay, so we have our raw file over here. Right, so Cutadapts let us do both the two things in a single command. So let's look at the syntax. Okay, so let's say our raw data is in this file called rawreads.fastq. We are going to build our command piece by piece. Okay, so let's begin. I will start cut adapt. After that, a let's say G A T C G G A G A G C. Okay, then Q. After that, let's go with 20. Then O. Let's call it clean. Clean reads already exists, so I will just call it clean dot fastq. Okay, and then input file is raw reads dot FastQ. Okay, so let us discuss this command actually. So first, let's break this down. So first, cut adapt, right? This code, cut adapt is the uh, command that will call the program. Then this portion, dash a and this sequence, this a flag stands for adapter. We paste the standard Illumina adapter sequence here. Cut adapt will hunt for this exact sequence in every read and snip it off. Then we have this command. Q flag stands for quality. This tells Cutadapt to chop off the ends of the reads if the Phred quality scores dips below 20. Okay, and then we have this command. What it is basically the O flag stands for output. This is the new clean file that we will be creating, and finally this is our raw input file that we will be entering. Okay, so let's hit enter and see what happens. Okay, and there you have it. Uh, because Cutadapt is highly optimized, it will chew through your FASTQ files in seconds, as you can see here. And let's see when Cutadapt finishes, it prints a beautiful summary report right here in your terminal. As you can see, this report is crucial for your lab notebook as well. So scroll up slightly in your terminal. you will see this section called total reads processed. Below that, it will tell you exactly how many reads contained adapters and how many base pairs were quality trimmed. Okay, so if you see that like 90% or more than that has of your reads had adapters trimmed, that means your biological DNA fragments were very short. And if it says something like you only had 2%, you 2% had adapters, your library preparation was very clean. Okay, so it will also tell you reads became too short after trimming, and by default, if Cutadapt trims a read down to like zero basis, it removes it entirely, and it keeps your final dataset perfectly clean. And just like that, you have professionally pre-processed next generation sequencing data. And so, if you were to run FastQC on your new clean reads.fastq file, those regex would magically turn into green check marks. Okay, so this data is now pure high quality biology. It is finally ready for the most computationally heavy task in all of bioinformatics, which is sequence alignment. So we will be tackling those portion in our next video. And so if this video helped you clean up your sequence pipelines, hit the like button and make sure you are subscribed, and put any errors you get in the terminal down in the comments and I will help you out. I'll see you in the next video.